moving place, all this stuff that we've talked about, pretty much all of it, takes place in the cytosol. The only exception was that first reaction of the pyruvate carboxylase that usually occurs in the mitochondria, and then it gets shuttled out to the cytosol. But pretty much all of that was in the glycolysis. So the glycolysis, gluconeogenesis, that's going to be going up here. So now the pyruvate that we generated is going to enter into the mitochondria, and we're going to really talk about two processes: pyruvate dehydrogenase, and then the citric acid cycle. That's going to involve the oxidation of those 3 carbons of pyruvate into carbon dioxide, right? And then those are going to create these high energy carriers, NADH and FADH2, that's going to go to electron transport, and then that's going to then be able to drive oxidative phosphorylation. We'll get into that in a day or two. All right, so what I want to do is give a couple of slides just to kind of show the importance of mitochondria in this oxidative process. So again, here's our mitochondria. And we're going to talk about, especially when we get to the, start talking about electron transport chain, we're going to get into the anatomy of the mitochondria in a little bit. It's a double membrane system, okay? So there's 2 lipid bilayers, right? So there's an outer mitochondrial membrane here. That outer mitochondrial membrane is relatively porous, and so things can pretty much get in and out of there at will. The tight membrane is this inner mitochondrial membrane, which is kind of convoluted in here. And then on the inside of that inner mitochondrial membrane is actually where the pyruvate dehydrogenase and the citric acid cycle within the mitochondrial matrix. And you could see the NADH and all that stuff is going to end up donating it to the electron transport chain, which actually lies in the inner mitochondrial membrane. So we're focused in the mitochondrial matrix.
Professor, are both of those membranes bilayers?
Both of them are bilayers, that's right, yes. All right, here's an electron micrograph of a muscle cell. And so here we have, what's this, mouse heart. So here we have actin and myosin fibers. So those are the protein fibers, the proteins that make up a muscle fiber that slide past each other and that consume a lot of ATP here. And what you see is nestled up right against them are some mitochondria, because those mitochondria are generating the ATP to basically allow those things to contract. Here's another example. You talk about, there's a lot of selection for motor activity, and that's in sperm flagella. So there's here's the flagella of a sperm. This is from bat, and then you notice so that that flagella is going to rotate right around. So there's basically a little molecular motor that consumes ATP to drive sperm motility, right? And there's going to obviously a lot of selection for sperm motility. And you see what's happened. See all the mitochondria that are just lined right up here, basically provide the ATP to help that drive that. That's bat sperm. Here's human sperm again. There's a whole bunch of mitochondria right here, just lined up basically to provide that molecular motor that drives the flagella in there. That's bat and human. It's not well known. Occasionally these hybridize, and then you get Batman sperm. All right, that joke kind of died. Anyway, so what we're going to do is we're going to go through and we're going to talk about the reactions. We're going to first take that pyruvate and we're going to talk about the reaction to the pyruvate dehydrogenase complex. So far, the enzymes that we've been talking about in glycolysis have been structurally relatively simple enzymes. Some of them are monomers, some of them are dimers. They tend to be globular proteins that work together. When we get into the pyruvate dehydrogenase complex, we're going to see a much more complicated tertiary and particularly quaternary structure of an enzyme. And while it's a kind of a single enzyme complex, it's actually made up of three different subreactions in a sense. One is known as pyruvate dehydrogenase itself. The second is known as dihydrolipoyl translase, or E2, and then the other one is this dihydrolipoyl dehydrogenase, E3. So what we're going to do is walk through that. The other thing that we're going to see as we go through here is that this reaction uses quite a few different coenzymes and prosthetic groups, other things, non-amino acid, non-polypeptide backbone stuff to help drive this reaction. And so we'll go through and we'll see those. And it's interesting, like this, I stole this from my daughter, this Monster Energy drink, right? If you look at the Monster Energy drink, you see what they put in there to make it, quote, energy drink. We're going to see some of this stuff. I'll point it out as we go through. The niacin that we're going to go through, that's actually part of NADH, right? That's what makes that nicotinamide ring. This pantothenic acid in here is one of those components in there. So we'll be able to see some of those types of things as we go through that. So, all right. So. This is a new outline, pyruvate dehydrogenase in the citric acid cycle.